Oxidative phosphorylation takes place in blank. Mitochondria. The outer membrane of a mitochondrion has pores that make it permeable to blank molecules and ions, but not to proteins. Small. The outer membrane of a mitochondrion has pores that make it permeable to small molecules and ions, but not to blank. Proteins. The convolutions of the inner membrane of a mitochondrion provide a very blank, large surface area. The inner membrane of a single liver mitochondrion may have more than 10,000 sets of blank and ATP synthesis molecules. Electron transfer systems. The inner membrane of a single liver mitochondrion may have more than 10,000 sets of electron transfer systems and blank. ATP synthesis. DNA and ribosomes of mitochondria differ from blank in the rest of the cell. Nuclear DNA and the ribosomes. Mitochondria reproduce within the cell more or less blank of the cell itself. Independently. Cells with high levels of blank may have thousands of mitochondria. Aerobic metabolism. Cells with high levels of aerobic metabolism may have thousands of mitochondria, whose cristae are more blank-packed than in less active cells. Densely. Cells with high levels of aerobic metabolism may have thousands of mitochondria, whose cristae are more densely packed than in less blank cells. Active. Electrons move through a chain of membrane-bound carriers spontaneously, driven by the blank and the relatively low reduction potentials of the various reduced substrates that undergo oxidation, e.g. TCA cycle in the mitochondrion. High reduction potential oxygen. Electrons move through a chain of membrane-bound carriers spontaneously, driven by the high reduction potential oxygen and the relatively low reduction potentials of the blank and the various reduced substrates that undergo oxidation. Inner membrane of the mitochondria is not permeable to anything that lacks a transporter. Transporter. Blank is the mechanism for transducing oxidative energy into ATP was first proposed by Peter Mitchell in 1961. Chemiosmosis. Chemiosmosis as the mechanism for transducing oxidative energy into ATP was first proposed by blank. In 1961, he was initially ridiculed, and then won the Nobel Prize for it. Peter Michel. The flow of electrons blank their energy hill to oxygen drives the uphill export of protons, establishing lower pH outside than inside. Down. The flow of electrons down their energy hill to oxygen drives the uphill export of protons, establishing blank outside than inside. Lower pH. The flow of electrons down their energy hill to blank drives the uphill export of protons, establishing lower pH outside than inside. Oxygen. The flow of electrons down their energy hill to oxygen drives the blank export of protons, establishing lower pH outside than inside. Uphill. Backflow down the pH gradient then drives blank. ATP formation. Backflow down the blank then drives ATP formation, later found to drive active transport, flagellar motion, nerve conduction and other processes. pH gradient. Electron carriers in two mitochondria, NADH, NADPH, FADH2. Electron carriers being small and blank, these can diffuse in from cytosol, also made directly in the mitochondrion. Water-soluble. Blank being small and water-soluble, these can diffuse in from cytosol, also made directly in the mitochondrion. Electron carriers. Electron carriers being small and water-soluble, these can diffuse in from cytosol, also made directly in the blank. Mitochondrion. Blank transfer 2 electrons. Nicotinamides. Blank transfer 1 or 2 electrons. Flavins. A blank is a single electron transferred whether it is one of a pair singly. Reducing equivalent. 3 types of carriers in the intermitochondrial membrane, blank, cychromes, and Phas proteins. Ubiquinone. 3 types of carriers in the intermitochondrial membrane, ubiquinones, blank, and Phas proteins. Cychromes. 3 types of carriers in the intermitochondrial membrane, ubiquinone, cychromes, and blank proteins. Phas. The electron carriers have a blank makes it soluble in the membrane. Hydrophobic tail. The electron carriers are small that it diffuses blank within the bilayer. Freely. Each prosthetic group of cytochromes consists of blank in a structure called porphyrin. 4 5-membered nitrogen-containing rings. Each prosthetic group of cytochromes consists of 4 5-membered nitrogen-containing rings nitrogens in a structure called porphyrin. Porphyrin. The blank of the prosthetic groups of cytochromes are coordinated with a central Fe2 or Fe3. Nitrogens. Blank is found in B-type cytochromes and in hemoglobin and myoglobin. Iron protoporphyrin 9. Blank is covalently bound to the protein of cytochrome C through thioether bonds to 2C-wise residues. Hemc. Blank is found in a type cytochrome has a long isoprenoid tail attached to one of the 5-membered rings. Hem-A. The porphyrin ring with its blank is the same in all cases, the groups hanging off the ring differ. Iron. Half reactions with blank standard potentials would be expected to pull electrons from those with lower potentials. Greater. Instantaneous blank could change the order, but it so happens that they don't. Concentrations. 2 methods, both depending on blank. All the carriers have spectra which differ enough that they can be distinguished. Absorption spectra. A solution in which some molecules of a carrier are oxidized and some are reduced will have an absorbance in blank the fully oxidized and fully reduced forms. Between. Method 1. Imagine a solution containing all the carriers. We apply an electron source but no oxygen to act as blank. Final acceptor. Method 1. All the carriers will become blank, and everything will stop at that point. Reduced. Method 1. Then we supply oxygen abruptly and watch the spectrum of each carrier. The first to become oxidized is blank to oxygen, the second precedes the first in the chain. Nearest. Method 2 determines the sequence of electron carriers, measures the effects of inhibitors on the blank of each carrier. Oxidation state. In the presence of an electron donor and O2, each inhibitor causes a characteristic pattern of blank carriers. Those before the block become reduced, and those after the block becomes oxidized. Oxidized, reduced. The mitochondrial electron transfer are made up of blank multiprotein complexes. Prosthetic groups, FMN, FAS, NADH dehydrogenase. Prosthetic groups add Phas 6 and 8 dehydrogenase. Prosthetic groups hemes Phas. Ubiquinone cytochrome C oxidoreductase. Prosthetic groups hemes cytochrome C. Prosthetic groups hemes CUA cub. Cytochrome oxidase. Separation of functional complexes of the respiratory chain. The outer membrane is first removed by treatment with the mild detergent digitonine. Inner membrane is then obtained by blank, and the fragments are gently dissolved in a second detergent. Osmotic rupture. Separation of functional complexes of the respiratory chain. The resulting mixture of inner membrane proteins is separated by blank into different complexes, and the enzyme ATP synthase. Ion exchange chromatography. Separation of functional complexes of the respiratory chain. The isolated complexes catalyze blank between donors, intermediate carriers, and oxygen. Transfers. Separation of functional complexes of the respiratory chain. In vitro, isolated ATP synthase has only blank, not ATP synthesizing, activity. ATP hydrolyzing. Complex I, NADH dehydrogenase, also called as blank. NADH ubiquinone oxidoreductase. The reaction couples NADH oxidation with blank coenzyme Q reduction. Complex I, it is also blank in that it moves protons in one direction, from inside the matrix to the space between the inner and outer membranes. Vectorial. Complex I, the space gains positive charge and it is called the blank. P-side. Complex I, the matrix becomes negative owing to the loss of H ions and is called the N-side. Negative. NADH ubiquinone oxidoreductase blank catalyzes the transfer of a hydride ion from NADH to FMN, from which two electrons pass through a series of Phas centers to the Phas center N2 in the matrix arm of the complex. Complex I. NADH ubiquinone oxidoreductase. The electrons transfer from N2 to Q on the membrane arm forms blank, which diffuses into the lipid bilayer. QH2. NADH ubiquinone oxidoreductase. This transfer of electron drives the expulsion from the matrix of 4 protons per pair of electrons. Expulsion. NADH ubiquinone oxidoreductase. Protein flux produces in blank across the inner mitochondrial membrane. Electron chemical potential. Complex 2, 6-onate dehydrogenase, path, 6-onate to fat, FADH2 to 3, 2Fe2S clusters to Q. Q. Blank, 6-onate dehydrogenase, 2 transmembrane subunits, C and D and 2 cytoplasmic extensions. Subunits A and B. Complex 2. Complex 2, 6-onate dehydrogenase, just behind the fat in subunit A is the binding site for 6-onate. Complex 2, 6-onate dehydrogenase, subunit B has blank phi S centers. 3. Complex 2, 6-onate dehydrogenase, ubiquinone is bound to blank, and hemib B is sandwiched between subunits C and D. Subunit B. Complex 2, 6-onate dehydrogenase, 2 blank molecules are so tightly bound to D that they show up in the crystal structures. Phosphatidylethanolammine. Complex 2, 6-onate dehydrogenase, blank move from 6-onate ultimately to Q. Electrons. Complex 2, 6-onate dehydrogenase, the hemla B is not on the main path but blank protects against the formation of reactive oxygen species by stray electrons. Supposedly. Complex 2, 6-onate dehydrogenase, QH2 from blank both diffuse freely within the bilayer, they enter complex 3. Complexes I and 2. Blank, cytochrome BC1. Complex 3. Complex 3, cytochrome BC1, since Q can transfer one electron at a time via a Q-free radical, it can blank the pairwise transfers from NADH and FADH2, passing electrons to the hema-containing carriers, which can take only one. Break. Complex 3, cytochrome BC1, a dimer of identical monomers each with blank different subunits. 11. Complex 3, cytochrome BC1, the core of each blank is 3 subunits, cytochrome B with its 2 hemes, 3 SKFES protein with its 2 Fe2S centers, and CYTC1 with its hema. Monomer. Complex 3, cytochrome BC1, the interface between monomers forms 2 caverns, each containing a QP site from one monomer and a QN site from the other. The Q intermediates move within these blank sheltered caverns. Blank, cytochrome oxidase, the final transfer to oxygen. Complex IV. Path of electrons through complex IV. The 3 proteins critical to electron flow are blank. Subunits I, II and III. Blank mitochondria can bypass complexes I through IV to generate heat. Plant cells keep the blank very low, so the second term is negative, and free energy is even more favorable than free energy not. NAD/NADH ratio. Most of the energy is used to pump protons out of the matrix, for transfer of one pair of electrons to oxygen. Complex I pumps 4 protons, complex III pumps another 4 protons, and complex IV pumps 2 protons. Energy. We have stored energy in the gradient. This proton motive force consists of blank due to the difference in concentrations between inside and outside, and the electrical potential energy due to the difference in charge between inside and outside. Chemical potential energy. Where is the site of oxidative phosphorylation in eukaryotes? Mitochondria. How many membranes does mitochondria have? 2. What are the membranes of the mitochondria? Inner and outer. What is the inner membrane of the mitochondria? Impermeable to most small molecules and ions and H. What is the outer membrane of the mitochondria? Freely permeable to small molecules and ions. What does the mitochondria consist of? Pyruvate dehydrogenase complex, citric acid cycle enzymes, fatty acid beta oxidation enzymes, amino acid oxidation enzymes, DNA, ribosomes, ATP, ADP, pi, Mg2, circa2, K, and soluble metabolic intermediates. For outer membrane, what is needed for the molecules to move freely? Porous. In inner membrane, what is needed to move through the membrane? Specific transporters.